The most cited scientific paper in history almost never existed because its author thought it was too routine to bother with. This is a biochemistry paper from 1951. Biochemistry being the study of the molecules that make living cells work. Its entire job was to answer one question: how much protein is in this sample? The same way a recipe asks how much flour before you can bake anything. That paper has accumulated 355,000 citations, more than Watson and Crick, more than Einstein on relativity. If you've ever received a vaccine or benefited from cancer research, this quiet little method was somewhere in the room. Oliver Lowry trained as both a physician and a research scientist, a combination that gave him biological curiosity and physicist-level precision. He spent time in Copenhagen working under Kai Linderstrom Lang, the gold standard of protein chemistry at the time, and it left a mark. By 1947, he was chairing the pharmacology department at Washington University in St. Louis, a post he'd hold for nearly 30 years. The person who made it work bench by bench was Nira Rosebrough, his technician, and by Lowry's own account, one of the best he'd ever had. 4 names on the paper, no one famous, just a careful team that cared more about getting it right than getting credit. The chemistry at the heart of this is almost embarrassingly simple. You add a copper salt to your protein sample, copper ions, the same element that turns old pennies green, and they latch onto the protein's backbone. Then you add the Folin Phenol Reagent, which reacts with that copper protein complex and turns the whole solution blue. The more protein you have, the deeper the blue, and a spectrophotometer, a device that measures how much light a liquid absorbs, gives you the exact number. Think of it like a pH strip from high school chemistry class, but instead of acidity, it's reading protein concentration. It was up to 100 times more sensitive than anything before it, capable of detecting protein in amounts measured in micrograms, millionths of a gram. The whole protocol fit on a few pages and required no equipment that wasn't already sitting in every lab. Here is the part that should humble every scientist. Lowry considered this a routine refinement of existing work, not a discovery, not worth a paper. He only wrote it up because colleagues kept asking him to share the protocol. The paper he almost didn't bother with became the most cited scientific document ever written. Because protein is everywhere in biology, the Lowry assay ended up everywhere too. Vaccine manufacturers used it to verify protein content in influenza vaccines, and it's still in the regulatory rulebook today. Cancer researchers used it to track signaling molecules, the chemical switches that tell cells when to grow, divide, or stop. Earl Sutherland's Nobel Prize-winning discovery of cyclic AMP, a molecular messenger inside cells, depended on knowing exactly how much protein he had. By 1987, this single paper was accumulating 10,000 new citations every year. In a database of 58 million scientific papers, fewer than 15,000 have ever crossed 1,000 total citations. Lowry's paper sits at 355,000. That's not a citation record, that's a different category of thing entirely. In the decades after publication, the Lowry assay quietly powered vaccine approvals, cancer research, and Nobel Prize-winning discoveries. Successor methods, the Bradford assay, then the BCA assay, eventually took over, but both are built on Lowry's copper chemistry. Lowry himself was genuinely puzzled by the paper's dominance. He never considered what he'd done a real discovery. The takeaway? The most powerful tools in science are not grand theories. They are reliable methods that anyone can pick up and use. The most cited paper in human history is essentially a recipe, and its author almost didn't write it down.